Today we are going to study angiosperms. Reproductive structures are born on a special structure called flower, hence they are called flowering plants. Yes, students, so this is how angiosperm is different from gymnosperm. Angiosperms are going to flower, a specialized organ which is going to produce male reproductive organ and female reproductive organ. Male reproductive structure is known as androecium or stamen, which releases pollen grains, and female reproductive structure is pistil or gynoecium, in which we find ovules. So ovules are later, after fertilization, they are going to develop into seeds which are enclosed by ovary. Nodri ovules ovary lintra enclose agyo, ovary after fertilization it is going to form fruit. So how these plants are? Usually these plants might be small and tiny. Okay, this smallest plant in angiosperm is Ulfia, and the tallest plant in angiosperm is Eucalyptus. About 100 meters in height it can grow. So these plants are not only helping us for the sake of food. Nodri bhalast angiospermic plants we consume them as a food, but it is also a fodder for cattle and used as a fuel and also for the purpose of medicine. So most of the angiosperms are having high medicinal values too. These angiosperms are divided into two main classes. One is dicotyledon and another one is monocotyledon. Dicotyledons andre ಎರಡು cotyledons irtav. In case of monocotyledon, only one cotyledon will be there. So ಯಾವ ಕಾಳು ತಗೋರಿ, ಯಾವ ಎರಡು ಬೇಳೆ ಆಗ್ತಾವಲ್ಲ, where you find two cotyledons. For example, gram. Okay. In case of monocotyledon, jawar, maize. These are having only one cotyledon. So not only with respect to cotyledon, we can also differentiate a plant with respect to leaf. Reticulate venation in dicot shows midrib, which branches to veins, veins branches to veinlets. Parallel venation in monocots, where veins and veinlets are arranged parallelly on leaf. In dicot you find tetra or pentamerous flower, trimerous flower in case of monocots. So now we are coming with male reproductive structure and female reproductive structure. Male reproductive structure is known as stamen or androecium. Female reproductive structure is known as pistil or gynoecium, which is having stigma at the terminal end, style, a long tubular style, and at the base swollen ovary which is having one or many ovules. Okay, the number of ovules may differ. In case of male, in case of stamen, that is male reproductive structure, we find anther and a long filament. Anther is going to produce pollen grain. Okay, now we are going to study how these pollen grains are produced. Idu section tagondiruvanta view. Okay, after taking the section of this anther, we are going to see this way, and how these pollen grains are developed, that we are going to study now. So when we are taking out the section, the sectional view is going to appear like this. There are many wall layers for the anther. Inside we find a sporogenous tissue. In that one cell I am going to take out. We call it as pollen mother cell. We call it as pollen mother cell, which is diploid. Up to here everything is diploid. Nodri stamen kuda diploid, flower kuda diploid, even plant is also diploid. Sporophyte ಅಂತಂದು ಹೇಳ್ತೇವೆ, same as that of gymnosperm. So when the pollen mother cell is diploid, now it is going to undergo reductional division, andre meiosis. It undergoes meiosis to give rise to a microspore tetrad. ಇದಕ್ಕೆ ಏನಂತೇವೆ ನಾವು? Microspore tetrad where all 4 microspores are arranged together. Now later they are going to develop into microspores. ಇವು microspores ಏನಿರ್ತಾವ್ರಿ haploid ಆಗಿರ್ತಾವ. So later these haploid microspore are going to develop into pollen grain. Now ಒಂದು microspore ಅನ್ನ ತೋರಿಸಿನಿ how it develops into a pollen grain. Okay, for this pollen grain we call it as male gametophyte. We call pollen grain here as a male gametophyte. Students, just try to compare the gametophytes from bryophyte, pteridophyte, gymnosperms and angiosperms. Okay, please study comparatively. So in case of here, pollen grains are called as male gametophyte, which are not free living. Okay, they are dependent on the sporophytic plant body, and when this anther is going to release pollen grain with the help of wind or some other agent, it reaches the stigma or the pistil. We call this process as pollination. ಇದಕ್ಕೇನಂತ ಹೇಳ್ತೇವೆ ನಾವು pollination ಅಂತ ಹೇಳ್ತೇವೆ. Okay, meanwhile we'll study the female reproductive structure, stigma, style and ovary. Ovary is going to contain numerous ovules. So when we are taking out one ovule here, it is going to appear this way. Okay, in this, how this embryo sac have been developed? This is a nucellus tissue. ಇದಕ್ಕೇನಂತೇವೆ ನಾವು nucellus tissue ಅಂತ ಹೇಳ್ತೇವೆ. Many numerous cells will be there. In that one cell, ಇಲ್ಲಿ ಮೊದಲಿಗೆ imagine ಮಾಡ್ಕೋಬೇಡ್ರಿ embryo sac ಇರೋದು. Only just imagine here only one cell is there. Okay, so how this from one cell this embryo sac has been formed, that we are going to study now. This one cell we are going to call it as megaspore mother cell, which is diploid. Okay, which is present in the nucellus. Same like pollen mother cell ille megaspore mother cell, which is diploid, which one cell from the nucellus is going to divide meiotically or undergoes reductional division to produce how many spores? 4 megaspores. ಏನಾಗ್ತದೆ, ಮೆಗಾಸ್ಪೋರ್ ಮದರ್ ಸೆಲ್ ಅಂಡರ್ಗೋಸ್ ಮಿಯೋಸಿಸ್ ಟು ಪ್ರೊಡ್ಯೂಸ್ 4 ಮೆಗಾಸ್ಪೋರ್ಸ್. ಅಂದರೆ ಇದು ಡಿಪ್ಲಾಯ್ಡ್ ಇರೋದು. ಮೆಗಾಸ್ಪೋರ್ ಏನು ಕೊಡ್ತದೆ ನಮಗೆ? ಹ್ಯಾಪ್ಲಾಯ್ಡ್. ವಿ ಗಾಟ್ ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಮೆಗಾಸ್ಪೋರ್. ಲೇಟರ್ ದಿಸ್ ಒನ್ ಹ್ಯಾಪ್ಲಾಯ್ಡ್ ಮೆಗಾಸ್ಪೋರ್, ವಾಟ್ ಇಟ್ ವಿಲ್ ಡೂ? It is going to form into an embryo sac. ಹಾಗಾದ್ರೆ other 3 they are going to degenerate. So all 3 can't develop at a time. So in nature only one can develop. So one gains that ability to develop into a embryo sac. How this one megaspore is going to develop into such a embryo sac, you will be studying it in PUC second year in detail. So students, now we are going to study the structure of embryo sac. Now I am putting an outline of for the embryo sac. Only this much is a embryo sac. Okay, if the wall layers are covering or integuments, if they cover an embryo sac, we call it as a ovule. ಈ structure ಗೆ ಏನಂತೇವೆ ನಾವು? ovule ಅಂತೀವಿ ಅಥವಾ megasporangia. We call it as a megasporangia when whole structure ಗೆ. So what all things it is having? Embryo sac ಒಳಗಡೆ ಏನೈತಿ? It is having synergids. 2 synergids are there. ಇದು ಒಂದು, ಇದೆರಡು. Okay, these are the synergids. What is this one? This is egg. So both together we call it as egg apparatus. Okay. In central, these 2 are haploid cells. ಇವೆರಡು haploid cells. They are going to fuse to form diploid polar nuclei. ಏನು ಇದು? ಪ್ಲಾಯ್ಡಿ. ಇದು ನೋಡ್ರಿ, ಎಗ್ಗಿಂದ ಪ್ಲಾಯ್ಡಿ, ಹ್ಯಾಪ್ಲಾಯ್ಡ್. ಸೈನರ್ಜಿಡ್ಸ್ ಇಂದು ಪ್ಲಾಯ್ಡಿ, ಹ್ಯಾಪ್ಲಾಯ್ಡ್. ಇದು ಒಂದು ಹ್ಯಾಪ್ಲಾಯ್ಡ್, ಇದೊಂದು ಹ್ಯಾಪ್ಲಾಯ್ಡ್. ಓಕೆ, ಬಟ್ ಹಿಯರ್ ಬೋತ್ ಹ್ಯಾವ್ ಬೀನ್ ಫ್ಯೂಸ್ಡ್ ಟು ಫಾರ್ಮ್ ಡಿಪ್ಲಾಯ್ಡ್ polar nuclei. So what are these? These are antipodal cells. Each cell is haploid here. They are not fused. Okay, so later on what happens? So this is the structure of ovule or megasporangia. This is what we call it as embryo sac. Embryo sac ಏನಂತೇವಿ ನಾವು? For embryo sac we call it as female gametophyte. So students, whenever I am using the term gametophyte means it is a gamete producing structure ಅಂತ ನೆನಪಿಟ್ಕೊಂಡ್ಬಿಡ್ರಿ. ಹಾಗಾದ್ರೆ ಫೀಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಏನು? ಫೀಮೇಲ್ ಗ್ಯಾಮೆಟ್ is egg. In case of male gametophyte, if it is a pollen grain, it produces 2 male gametes. Okay, ಎರಡು ಮೇಲ್ ಗ್ಯಾಮೆಟ್ ಇಲ್ಲೇ ಇರ್ತಾವು, but always they tend to remain at the tip ಒಳಗಡೆ ಇರ್ತಾವೆ. ಯಾಕೆ? I'll tell you now. So what happens after pollination? Pollen grains reaches the stigma. So pollen grain is huge. It can't travel through the tissues of stigma or style. Slowly it gives rise to a pollen tube. ಏನು ಕೊಡಾತದ್ರಿ ಇದು? Pollen tube ನ ಕೊಡ್ತದೆ. Ultimately it reaches one ovule. Okay, so this is how I am going to draw a ovule and show you people. Okay, how the pollen tube reaches? See, now it has come here. Now it has reached the ovule. So it will be having 2 male nuclei. Soon after what happens? Soon after it enters, the synergid cells are going to degenerate. One fuses with egg. Egg cell jodi fuse ಆಗ್ತದೆ. After fusing it forms diploid zygote. The process of fusing ಏನಂತೇವೆ ನಾವು? Syngamy. For the process we call it as syngamy. It gives rise to a diploid zygote. Zygote, what it does? Later it forms embryo. Embryo ಆಗಿ develop ಆಗ್ತದೆ. See, embryo is already here. I have drawn the image of embryo. Later, ಇನ್ನೂ ಒಂದು ಮೇಲ್ ನ್ಯೂಕ್ಲಿಯೈ ಅದ. Another male nuclei, what it does? It fuses with the polar nuclei. But already the polar nuclei is diploid. So what it does? It forms triploid primary endosperm nuclei. It forms the full form primary endosperm nuclei. Later this develops into endosperm. So students, always remember endosperm in case of angiosperm is a post-fertilization product. It is very important as for the NEET or CET aspirants. Post-fertilization andre after fertilization endosperm develops in case of angiosperms. But in case of gymnosperms, ಈ ರೀತಿ 2 ಮೇಲ್ ನ್ಯೂಕ್ಲಿಯೈ ಇರುವುದಿಲ್ಲ gymnosperms ನಲ್ಲಿ. So soon after when it fuses with egg, it forms a zygote only. But ಇಲ್ಲಿ endosperms ಅಲ್ಲಿ ಏನಾಗಿರ್ತದೆ? It keeps on dividing. Haploid ಇರ್ತದೆ endosperm. ಅಲ್ಲೇ polar nuclei ಅಂತ ಇರೋದಿಲ್ಲ. So endosperm is a pre-fertilized product in gymnosperm. In case of angiosperm, it is a post-fertilization product which is triploid. Nodri, after fertilization we got egg developed into embryo, polar nuclei developed into endosperm, and ovule after fertilization it develops into seed, and ovary develops into fruit. Students, please don't forget to subscribe my channel. Further, I will also be explaining you morphology on flowering plants. Thank you.